So up to this point, we've discussed DNA, RNA, and proteins. So these components are the basis of molecular diagnostics and molecular techniques. Chapter 4, we discussed extraction methods of both DNA and RNA. So now Chapter 5 is covering resolution and detection of nucleic acids. So this is done in several ways. So these are some of the things we'll be discussing in Chapter 5. The objectives we'll cover are explaining the principle and performance of electrophoresis as it applies to nucleic acids, comparing and contrasting agarose and polyacrylamide gel polymers, explain the principles and performance of capillary electrophoresis as it applies to nucleic acid separation, describing the general types of equipment used for electrophoresis, discussing methods and applications of pulse field gel electrophoresis, and comparing and contrasting detection systems used in nucleic acid applications. So we've already touched on electrophoresis, but here we'll go into it in a little more detail. So electrophoresis is the movement of molecules by an electric current. So nucleic acids move from a negative to a positive pole. It can occur in a solution, but it's practically done in a matrix to limit migration and contain the migrating material. Electrophoresis is routinely applied to the analysis of proteins and nucleic acids. Each phosphate group of DNA polymer is ionized, making DNA a negatively charged molecule. So under an electric current, as mentioned before, DNA will migrate toward the positive pole, the anode. When DNA is applied to a macromolecular cage such as agarose or polyacrylamide gel, its migration under the pole of the current is impeded depending on the size of the DNA and the spaces in the gel. Because each nucleotide has one negative charge, the charge-to-mass ratio of molecules of different sizes will remain constant. DNA fragments will therefore migrate at speeds inversely related to their size. So on this slide we have three examples of electrophoretic patterns. The movement of the molecules are impeded in the gel, so the molecules will collect or form a band according to their speed of migration, and the concentration of the gel or buffer in use affects the resolution of the fragments of different size ranges. So here you can see we have a 2%, a 4%, and a 5% agarose gel. So here on the 2% you can see that the molecules move faster and they move closer to the bottom. The 4% are impeded a little more, and then the 5% are impeded the most, where the molecules stay closer to the application region. When performing electrophoresis, it can be performed in tubes, slab gels, or capillaries. Slab gel electrophoresis can be either in horizontal or vertical format. The samples are introduced into the wells at the top of the gel, and then they move towards the positive anode. Very large DNA molecules are separated by pulsed field gel electrophoresis. Pulsed field gel electrophoresis, also known as PFGE, is a technique that separates pieces of DNA in size of 50,000 to 250,000 base pairs that can't be resolved efficiently by simple agarose electrophoresis. Even in the lowest concentration of agarose, very large fragments are too severely impeded for correct resolution. Limiting mobility is reached when a DNA molecule is of such a size that it can move only lengthwise through the successive pores of the gel. Pulses of current applied to the gel in alternating dimensions enhance migration for very large DNA molecules, and this is the pulse field gel electrophoresis technique. There are several types or variations of pulse field gel electrophoresis. Field inversion gel electrophoresis, also known as FIGE, is probably the easiest to perform with a minimum of special equipment. The only requirement besides a regular gel electrophoresis box and a power supply is a device that enables periodic inversion of the electric field direction over the course of the experiment. This method was derived from experiments done on modified orthogonal field alternation gel electrophoresis. It was based on the observation that obtuse angles lead to a better separation, the widest angle being 180 degrees. The 4 electrodes pulse field gel electrophoresis system was reduced to a standard submarine gel electrophoresis box with only 2 electrodes. This simple configuration generates a highly uniform electric field across the gel, making the lane-to-lane comparison very easy. Another type, transverse alternating field electrophoresis, TAFE, refers to a pulse field system that uses a vertical gel and a simple electrode geometry. The angle between A and B fields is not constant down the length of the gel. At the wells, it's a 115-degree angle, but it's much greater at increasing distances from the well. Contour-clamped homogeneous electric field uses alternating polarity in an electrode array, and rotating gel electrophoresis uses rotating gel with fixed poles to perform the procedure. So this slide just shows a visual of the different types of pulse field gel electrophoresis. So the first one, PFGE, or the field inversion gel electrophoresis, has alternating positive and negative poles. Transverse alternative field electrophoresis has transverse angle orientation of poles on a vertical gel. The contour-clamped homogeneous electric field, CHEF, has the alternating polarity in an electrode array. So here you see the positive, the negative, the positive, the positive, the negative. And then finally, the rotating gel electrophoresis has rotating gel with fixed poles. So you can see here. Polyacrylamide gel electrophoresis, also known as PAGE, is a method that very small DNA fragments and single-stranded DNA are best resolved. Acrylamide in combination with cross-linker methylene bis acrylamide are used. The methylene bis acrylamide polymerizes into a matrix that has consistent resolution characteristics. Polyacrylamide was originally applied mostly for protein separation, but now is routinely used for nucleic acid analysis for sequencing, mutation analysis, nuclease protection assays, and other applications that require high resolution of nucleic acids. Unlike agarose gel that polymerizes upon cooling, polyacrylamide requires the use of a catalyst, either ammonium persulfate, APS, plus NN and tetramethylethyldamanine, also called TMED, or a light activation. So for those of you who have already gone through your chemistry rotations and have done the protein electrophoresis on Sebia, that's an example of a ready-to-use agarose gel. The main advantage of polyacrylamide over agarose is the high resolution for small fragments. So the variation of one base pair in 11 kilobyte molecule can be detected. Capillary electrophoresis separates solutes by charge-mass ratio. The widest application of this type of electrophoresis has been in the separation of organic chemicals in pharmaceutical industry, and also the separation of carbohydrates. It's also been applied to the separation of inorganic ions and metal ions. Capillary electrophoresis has the advantage of faster analytical runs and lower cost per run. Increasingly, it's being used for separation and analysis of nucleic acids as well. So in this type of electrophoresis, the analyte is resolved in a thin glass capillary. The capillary is usually 30 to 100 centimeters by 25 to 100 micrometers internal diameter. Fused silica is used as the capillary tube because it's the most transparent material, allowing for the passage of fluorescent light. The fused silica is then covered with a polyimide coating for protection, except for an uncoated window where the light is shined on the fragment as they pass a detection device. As mentioned before, the separation is based on size, and also capillary gel electrophoresis is a more rapid, automated technique than slab gels, and it runs at a higher charge per unit area. The sample is introduced into the capillary through either electrokinetic, hydrostatic, or pneumatic injection. Electrokinetic injection is used for nucleic acid analysis. With electrokinetic injection, the platinum electrode close to the end of the capillary undergoes a transient high positive charge to draw the sample into the end of the capillary. When the current is established, the injected nucleic acid migrates through the capillary. Nucleic acid resolution by capillary electrophoresis is used extensively also in forensic applications. It has other applications in the clinical lab, such as clonality testing, microsatellite instability detection, and bone marrow engraftment analysis. So now that we've discussed an overview of the different types of electrophoresis, we're going to talk about the components of the electrophoresis procedure or methods. So the first important component are buffers. So buffers carry current and protect samples during electrophoresis. It's accomplished through the electrochemical characteristics of the buffer components. So a buffer is a solution of a weak acid and its conjugate base. The pH of a buffered solution remains constant as the buffer molecules take up or release protons given off or absorbed by other solutes. Aside from protecting the sample from damage, the buffer also prevents severe fluctuation in the pH of the gel. In order for nucleic acids to migrate properly, the gel must be immersed in buffer that conducts the electric current efficiently in relation to the buffering capacity. Common buffers used for DNA electrophoresis are tris-borate EDTA, tris-acetate EDTA, and tris-phosphate EDTA. Examples of RNA electrophoresis buffers are 10-sodium phosphate and MOPS buffer. So these type of buffers have different properties than those used for DNA electrophoresis. Buffer additives are used to modify the sample molecules. Such additives are formaldehyde or urea. So these are denaturing agents, typically used with RNA electrophoresis. The next component of electrophoresis, one that probably is the most important, is the electrophoresis equipment itself. So gel electrophoresis is performed in one of two conformations, horizontal or vertical. So in general, agarose gels are run horizontally and polyacrylamide are run vertically. Horizontal gels are run in acrylic containers called gel boxes or baths that are divided into two parts with a platform in the middle on which the gel rests. Because the gel is submerged throughout the loading and electrophoresis process, horizontal gels are sometimes referred to as submarine gels. A red connector is attached to the positive outlet on the power supply and a black to the negative port on the two separate compartments of the electrophoresis equipment. Vertical gel boxes have separate chambers connected by the gel itself. The electrodes are attached to the upper and lower buffer chambers to set up the current that will run through the gel. The gel must be clamped in place before filling the upper chamber with buffer. The positive electrode will be in contact with the bottom of the gel in the buffer. The negative electrode will be in contact with the top of the gel and a separate buffer compartment on the top of the insert. A component of the electrophoresis equipment are the combs. These are used to put wells in the cast gel for sample loading. The number and size of the comb's teeth determine the number of wells in the gel and the sample volume that can be added to each well. There's the regular comb, in which the wells are separated by an ear of gel, and the shark's tooth comb, where the wells are immediately adjacent. Advantages to the shark tooth configuration is that the lanes are immediately adjacent to one another, aiding in lane-to-lane comparison. The comb can also be left in place with the shark's tooth, whereas the standard comb or the regular comb needs to be removed before loading sample. Another important component of electrophoresis is the use of the proper gel concentration for sample size range. Small pieces of DNA, those measuring 50 to 500 base pairs, are resolved on more concentrated agarose gels, for example, 2 to 3% agarose gel. The lower the agarose concentration, the larger the size of the DNA fragments that can be resolved. This is due to high concentrations impeding movement or migration for smaller sizes, so they don't allow all to travel quickly to the end of the gel, while very low concentrations of agarose produce weak gel that can be easily broken and allow larger sized fragments to move through the gel without getting stuck. The proper comb and gel size also must be considered, since they are dependent on the size of the fragments to be resolved. Prior to loading the sample containing isolated nucleic acid onto the gel, tracking dye and a density agent are added to the sample to aid in monitoring the progress of electrophoresis. The dyes migrate at specific speeds in given gel concentration and usually run ahead of the smallest fragments of DNA. They are not associated with the sample DNA, so they don't affect its separation. The density agent is added to increase the density of the sample compared to the buffer, so then the sample sinks into the well instead of diffusing into the buffer. Detection of nucleic acid bands can be done by staining during or after electrophoresis. Dyes that specifically associate with nucleic acid are used to visualize the different sizes of nucleic acids after or during electrophoresis. Stain bands can be observed on the gel after the procedure is complete. The agents used most frequently are fluorescent dyes. Ethidium bromide is used commonly for double-stranded DNA. Cyber green or cyber gold are used for single or double-stranded DNA or for RNA. And silver stain is a more sensitive stain used for single or double-stranded DNA, for RNA, and for proteins. Section on resolution and detection of nucleic acids. So in summary, electrophoresis is used to separate molecules by size and or charge. Nucleic acid fragments can be resolved on agarose of polyacrylamide gels. Pulse field gel electrophoresis is used to resolve very large DNA fragments. Capillary gel electrophoresis is more rapid and automated than slab gel electrophoresis. And finally, the choice of electrophoresis method depends on the type and size of sample that you're analyzing.